What is pKa? pKa is a negative base to the minus 10 logarithm of the acid dissociation constant of a solution. This is represented by the circled formula. Dissociation is a process in which molecules split into smaller molecules. The pKa value indicates the strength of an acid. The lower the pKa value, the stronger the acid. This formula shows that pKa and pH are equal when half of the acid has dissociated. This graph represents the pKa value of the amino acid glycine. Amino acids are organic compounds that combine to form proteins. The human body uses proteins to break down food and to grow and repair body tissue. Proteins are formed from polypeptides, which are polymers made from various amino acids. An amino acid consists of 5 components. It contains a carboxylic acid, an amino functional group, a hydrogen, and an O group that are all attached to a tetrahedral carbon atom. This carbon is known as the alpha carbon. An O group of an amino acid is what distinguishes one amino acid from another. This is the structure of the amino acid glycine. There are 20 different types of amino acids, which are categorized as essential, non-essential, polar and non-polar amino acids. Essential amino acids cannot be synthesized by the body and therefore must be obtained from the diet. There are 9 essential amino acids. Non-essential amino acids are produced by the body, even if we do not receive them from the food we consume. There are 11 non-essential amino acids. Amino acids are further characterized as being acidic, basic, polar or non-polar. Acidic amino acids are negatively charged at physiological pH, whereas basic amino acids are positively charged at physiological pH. Non-polar amino acids are hydrophobic, meaning they are water insoluble and will repel an aqueous environment. They reside in the interior of proteins. Polar amino acids are hydrophilic, meaning they are water soluble and can therefore interact with an aqueous environment. They are located on the exterior surface of proteins. Primary structure of amino acids. These are basic peptide bonds and the most basic form of protein. We see lysine and glycine bond here via hydrogen bonds. They have an NC terminal bonding amino and carboxyl groups. These bonds are flexible, although well defined. The sequence can vary and is flexible overall. Structure often consists of an alpha helix or beta sheet structure. For an alpha helix, the proteins at this stage form into an obvious helix structure. This is known as local folding. This occurs because the hydrogen in the protein bonds to different amino acids in the polypeptide chain backbone. Amino acid side chain are not involved in this because they extend away from the axis of the amino acid. The actual folding occurs between the imide hydrogen of one amino acid and the carboxyl oxygen of another amino acid.
Tertiary structure takes many forms. It has overall folding of a single polypeptide chain. In this case, side chains stick structures to one another, hydrogen bonds, van der Waals interactions, hydrophobic interactions, salt bridges and disulfide bonds all work together to achieve overall structure of the protein. Quaternary structure is the most complex and complete form of amino acid folding. An example of this would be hemoglobin. It's a tetramer and consists of 2 different subunits made of 2 different dimers, alpha beta 1 and alpha 2 beta 2.